channel. This is Dr. K. Srinivas Reddy, Assistant Professor of Botany, Government Degree College for Women, Nalgonda. In this video, I am going to discuss about the topic nucleus. This topic is the part of your fifth paper, that is cell biology and genetics, which is in fifth semester. Let us come into the topic. The topic which we are going to discuss under four subtopics. They are introduction, morphology, structure of the nucleus, and functions of the nucleus. Under morphology, we are going to discuss occurrence and position of the nucleus, number of nuclei present per each cell, and shape of the nucleus, and size of the nucleus. Under structure of the nucleus, we are going to discuss about four main parts of the nucleus. They are nuclear membrane, nucleoplasm, chromatin fibers, and the nucleolus. Let us coming to the introduction part of the nucleus. Nucleus is the one of the important cell organelle. It is considered as the heart of the cell. In almost all the cells, DNA is confined, replicated, and transcribed in the nucleus. DNA is present inside the nucleus and which is replicated in the nucleus and transcribed in the nucleus. Replication means production of daughter DNA molecules from the parent DNA, and transcribed means formation of mRNA or transcription of mRNA from the DNA. Replication and transcription, both processes takes place inside the nucleus. So here DNA present inside the nucleus, replication and transcription takes place in the nucleus. The nucleus thus controls different metabolic activities as well as hereditary activities of the cell. Nucleus, which controls different metabolic activities of the cell and hereditary activities of the cell. Hereditary means the transformation of traits or characters from parental generation to offsprings. Nucleus is also known as karyon. Karyon is a synonymous word in Greek language for this cell organelle. The nucleus was discovered and named by Robert Brown in 1835 in plants. Robert Brown discovered nucleus in orchids. The role of nucleus in hereditary was established by the grafting experiments of Hammerling in 1953 with Astabularia. Hammerling conducted grafting experiments in Astabularia. Based on his research findings, he concluded that nucleus play a vital role or important role in heredity. Coming to the morphology. Morphology will be discussed under four headings. They are occurrence and position, number, size, and shape. First, we are going to discuss about occurrence and position. The nucleus is present in all eukaryotic cells, but it is absent in mature sieve tubes and erythrocytes or RBCs. Even though nucleus is present in young sieve tubes and RBCs, at maturity they lose the nucleus. In prokaryotes, cells do not have tree nuclei. The genetic material is called nucleide. The genetic material in the prokaryotic cells is called nucleide or genopore. In prokaryotes, the genetic material which is freely distributed in the cytoplasm, where the genetic material is not confined by a particular membrane. And coming to the position of the nucleus, the position of the nucleus is often variable. Usually, nucleus remains located in the center of the cell, but in highly differentiated cells, nucleus is pushed to one side of the cell. Highly differentiated means matured cells. In matured cells, the nucleus is pushed to one side of the cell due to formation of a large vacuole. Usually, in young cells, more number of small nuclei are present, but in matured cells, all these small vacuoles merge to form a large vacuole. This vacuole is present at the center. As a result, the nucleus is pushed a side of the cell. Coming to the second heading, that is number of nuclei. Based on the number of nuclei, cells may be uninucleate, binucleate, sinusotic or syncytial. Uninucleate, binucleate, sinusotic or syncytial. Uninucleate. Usually, only one nuclei is present in each cell. This condition is called uninucleate. Most of the cells possess only one nuclei. Binucleate. The cells which possess two nuclei are known as binucleate cells. For example, if you take tapetal cells of Euphorbiaceae, these cells possess two nuclei per cell. And also some of the liver cells are binucleate. Sinusotic or syncytial. Presence of more than two nuclei is called sinusotic. In case of plants, it is called sinusotic condition. In case of animals, polynucleated condition is called syncytial condition. Sinusotic. The plant cells which possess more than one nuclei are called sinusotic cells. Example, Vaucheria, which is an algae, Mucor is fungi, and some cells of endosperm of seeds. Endospermal cells contain more than two nuclei per cell. When come to animals, osteoblasts contain more than one nuclei. So these cells are called syncytial cells. Osteoblasts are syncytial cells. Polykaryotes of bone marrow. Osteoblasts are the polykaryotes of bone marrow. Let us come into the shape of the nucleus. Shape of the nucleus is mainly depending on cell shape and function. Here you can observe a table. In the first column, I have given the different shapes of cells, and in the second column, the shape of the nucleus which the cells possess. Here you can see spheroidal cells, cuboid cells, and polyhedral cells. Spheroidal cell, cuboidal cells, and polyhedral cells possess spheroidal shaped nucleus. Here you can see in the table. Spheroidal cells, cuboid cells, polyhedral cells possess spheroidal nucleus. And coming to the second one, cylindrical cells, prismatic cells, or fusiform cells. Cylindrical cells, prismatic cells, or fusiform cells possess ellipsoidal shape nucleus. Ellipsoidal shaped nucleus is present in the cylindrical shaped cells, prismatic cells, and fusiform cells. Next, squamous epithelial cells possess discoidal nuclei. Discoidal nucleus. Leukocytes are the white blood cells, possess horseshoe-shaped nuclei. In this way, different shaped cells possess different shaped nuclei. The shape of the nucleus mainly depends on the shape of the cell. Next, coming to the size of the nucleus, generally, nucleus occupies about 10% of the cell volume. Nucleus occupies about 10% of the value. Nuclei vary in size from 3 micrometers to 25 micrometers in diameter, which is mainly depend on cell type. The size of the nuclei vary from 3 micrometers to 25 micrometers in diameter depending on the cell type. The nucleus size has a constant relation to the cytoplasmic volume, which is expressed in the name of the nucleoplasmic index. The size of the nucleus has a constant relation to the cytoplasmic volume, which is known as nucleoplasmic index, that is known as NP, nucleoplasmic index. The size of the nucleus has a constant relation to the cytoplasmic volume of that particular cell, which is known as nucleoplasmic index, or you can say NP. Here we can calculate the size of the nucleus by using the following formula, that is NP is equal to Vn by Vc minus Vn. NP is equal to Vn by Vc minus Vn, where NP is nucleoplasmic index and Vn is the volume of the nucleus. Here Vc is the volume of the cytoplasm, and here Vn is the volume of nucleus. With the help of this formula, one can measure the size of the nucleus of the cells. The size of the nucleus has a constant relation to the cytoplasmic volume of that particular cell, which is known as nucleoplasmic index, or NP. We can measure the size of the nucleus of a particular cell by using a formula, which is NP is equal to Vn by Vc minus Vn, where NP is equal to nucleoplasmic index and Vn is the volume of nucleus and Vc is the volume of cytoplasm, and the Vn is the volume of nucleus. With the help of this formula, one can measure the size of the nucleus of a particular cell. Let us discuss about the structure of nucleus. The nucleus is composed of 4 main parts, namely nuclear envelope, nucleoplasm, chromatin fibers, nucleolus. These are the 4 main parts of nucleus. Number 1 is nuclear envelope or karyotheca. Second one, nucleoplasm or karyolymph, and chromatin fibers or chromatin network, nucleolus. So these are the 4 main parts of the nucleus. Here you can see these 4 parts in the picture very clearly. This is the envelope of the nucleus, this part, which contain 2 membranes, outer and inner membranes, and inside the membrane there is a network of fiber. This is called chromatin fiber, and here you can see the pink colored part, rounded part, which is nucleolus, and nucleoplasm is also present inside the nucleus. So these are the main parts: nuclear envelope, nucleoplasm, chromatin fibers, and nucleolus. Let us discuss about these parts in detail. First, we are going to discuss about the nuclear envelope or karyotheca. In all eukaryotes, the nucleus is surrounded by an envelope which is known as nuclear envelope. In all eukaryotic cells, the nucleus is surrounded by an envelope which is known as nuclear envelope or karyotheca. The nuclear envelope separates the nucleoplasm from the cytoplasm. Nuclear envelope separates the nucleoplasm, which is present inside the nucleus, and this nucleoplasm separates from the cytoplasm by nuclear envelope. Nuclear envelope is composed of two unit membranes which are made up of lipoproteins. Nuclear membrane consists of two unit membranes, namely one is inner membrane and second one is outer membrane. Here you can see in the picture, this one is the inner membrane, and this one is the outer membrane. Nuclear membrane composed of two unit membranes. This is inner unit membrane. This one is the outer unit membrane. Usually inner membrane is smooth in nature, whereas outer membrane is rough in nature due to the presence of ribosomes. And also the outer membrane is extended into our endoplasmic reticulum, our rough endoplasmic reticulum. Here you can see this is the endoplasmic reticulum. Endoplasmic reticulum is connected with the outer membrane of the nuclear envelope. And here you can see the each membrane, inner as well as outer membranes, is about 75 to 90 angstroms in thick. Both are unit membranes, both are lipoprotein membranes, and the thickness of the membranes is about 75 to 90 angstroms. 75 to 90 angstroms. These two membranes, inner and outer membranes, are separated by a space or empty space, you can say. There is a space in between inner and outer membrane. This space is called perinuclear space. Perinuclear space. It is about 100 to 150 angstroms. The space which is about 100 to 150 angstroms. The gap in between these two membranes is about 100 to 150 angstroms. This place, the empty space which is present in between these two membranes, is called perinuclear space. Perinuclear space. This space, which is appeared in yellow color in the picture, this space is called inner perinuclear space. Perinuclear space. And you can see the small apertures present in the nuclear envelope. These apertures are called nuclear pores or nuclear pore complexes. Ultrastructure of the nuclear pore complexes will be discussed in next video. Okay, so the pores which are present in the nuclear envelope is called nuclear pore complex. The pores present in the nuclear envelope or karyotheca are called nuclear pore complexes. The ultrastructure of the nuclear pore complexes will be discussed in my next video. Okay. Nuclear envelope or karyotheca. In all eukaryotic cells, the nucleus is surrounded by an envelope which is known as nuclear envelope. This nuclear envelope separates the nucleoplasm from the cytoplasm. Nuclear envelope composed of two unit membranes. One is inner membrane and the second one is outer membrane. Both membranes are unit membranes and made up of lipoproteins. And each membrane is about 75 to 90 angstroms in thick. And these two membranes are separated by a space of 100 to 150 angstroms. This membrane space, this intermembrane space, is called perinuclear space. In the nuclear envelope, many apertures are present. Our nuclear envelope is perforated by many apertures. These apertures are called nuclear pores or nuclear pore complexes. Okay. Next, we will discuss about nucleoplasm. Let us discuss about the nucleoplasm. Nucleoplasm is also known as nuclear sap. and it is also known as karyolymph, nucleoplasm or nuclear sap or karyolymph. The interior of the nucleus is filled with a transparent, granular, homologous fluid. The interior of the nucleus is filled with a transparent, granular, homologous fluid. It is called as nucleoplasm. The fluid which is present in the nucleus is called nucleoplasm. Nucleoplasm is slightly acidophilic in nature. Acidophilic in nature. The chromatin fibers and nucleolus remain suspended in the nucleoplasm or karyolymph. Chromatin fibers and nucleolus remain suspended in the nucleoplasm. And if you see the chemical composition of the nucleoplasm, nucleoplasm is mainly composed of nucleoproteins and also contains inorganic and organic substances. For example, nucleic acids, proteins and enzymes. These are the organic substances which are present in the nucleoplasm. Minerals are the inorganic substances. The most important enzymes in the nucleoplasm are the DNA polymerase, RNA polymerase, NAD synthetase, alkaline phosphatase, and dipeptidase. Dipeptidase. The most important enzymes, nuclear enzymes present in the nucleoplasm are the DNA polymerase, which is involved in DNA synthesis, RNA polymerase, which is involved in the formation of mRNA from the DNA during transcription, NAD synthetase, which is involved in the synthesis of NAD, alkaline phosphatase, and dipeptidase. And events which are taking place in the nucleoplasm are replication of DNA, transportation of RNA, transport of materials, formation of spindle fibers. So these are the main events which are taking place in the nucleoplasm. Replication of DNA. What is replication? Production of two daughter molecules from one parental DNA is called replication, which is taking place in the DNA, which is taking place in the nucleus. And transportation of RNA. Usually mRNA, rRNA, all types of RNAs are synthesized inside the nucleus. These RNAs are transported to cytoplasm from the nucleoplasm. And transport of materials like proteins, enzymes, and other inorganic materials are transport from cytoplasm to nucleoplasm and nucleoplasm to cytoplasm through nuclear pore complexes. Formation of spindle fiber during cell division, which is also one of the event which is taking place in the nucleoplasm. So nucleoplasm is also known as nuclear sap or karyolymph. The fluid which is present inside the nucleus is called nucleoplasm, which is acidophilic in nature. Chromatin fibers and the nucleolus remain suspended in the nucleoplasm. Nucleoplasm mainly composed of nucleoproteins and also contains inorganic and organic substances. Organic substances like nucleic acids, proteins, enzymes, and minerals are the inorganic substances. The most important enzymes which are present in the nucleoplasm are the DNA polymerase, RNA polymerase, NAD synthetase, alkaline phosphatase, and dipeptidase. Events which are taking place in the nucleoplasm are replication of DNA, transportation of RNA, transportation of materials, formation of spindle fibers. Okay. Let us coming to the chromatin fibers. Elongated thread-like structures present in the nucleoplasm are known as chromatin fibers. Thread-like, coiled, and elongated structures present in the nucleoplasm are known as chromatin fibers. These chromatin fibers absorb basic stains like the basic fusin, and the chromatin fibers can only absorb in the interphase nucleus only. During division phase, these chromatin fibers condensed into chromosomes. During division phase, these chromatin fibers condensed into chromosomes, so that we can observe these chromatin fibers during interphase nucleus only. Chemically, chromatin fibers are nucleoproteins. Chemically, chromatin fibers are nucleoproteins. It means chromatin is made up of DNA and proteins. Here, two types of proteins are present in the chromatin. Number one is histone proteins and second one is non-histone proteins. Histones and non-histones. First, we are going to discuss about histones. The histones are small basic proteins, basic in nature, because they have highly content of basic amino acids such as arginine and lysine. Due to the presence of more amounts of arginine and lysine, histones are basic in nature. Being basic, histones bond tightly with DNA, which is an acid. DNA is an acid. Histones are proteins which are basic in nature. So as a result, these histone proteins bind tightly with DNA molecule. Histone proteins lower the genetic activity of DNA or depress the genetic activity of DNA and involved in packing of DNA molecule into chromatin fiber. Histone proteins lower the genetic activity of the DNA molecule and involved in packing of DNA molecule into chromatin fibers. Coming to the non-histone proteins, non-histones are acidic in nature. These proteins either present in the nucleoplasm or in the chromatin. Non-histones either present in the nucleoplasm or in the chromatin fiber. These proteins concerned with gene expression. These proteins play an important role in the gene expression. Gene expression is nothing but production of protein. When a gene is expressed in the form of protein, so it plays an important role in the gene expression. RNA polymerase is the best example for non-histone protein which is present in the nucleoplasm. The chromatin fibers are either distributed loosely, or you can say uncoiled, or form variously shaped dense clumps, or you can say coiled. The chromatin fibers either distributed in uncoiled manner or coiled manner. When you stain these chromatin fibers, which appear in two ways, some regions of the chromatin fibers appear in pale yellow color, and in some other regions chromatin fibers appear in dark color. The chromatin fiber which appears in pale color are called euchromatin, where the chromatin loosely coiled and take less stain, and the chromatin where darkly appeared, where the chromatin coiled more and take high amount of strain, as a result which appear in dark color. This chromatin is called heterochromatin. Euchromatin contain more amount of active DNA and heterochromatin comparatively less amount of DNA in relation to RNA. Euchromatin contain more amount of active DNA, whereas heterochromatin contain DNA which is inert in nature, which is not genetically inactive. Okay. Next, we are going to discuss about the nucleolus. Let us coming to the nucleolus, which is the last part which we are going to discuss. The large spherical body present in the nucleoplasm is known as nucleolus. The large spherical body which is present in the nucleoplasm is known as Nucleolus. The nucleolus was discovered by Fontana in the year 1774. Coming to the size of the nucleolus is found to be related with the synthetic activity of the cell. The size is mainly depends on the synthetic activity of the cell. Therefore, the cells which synthesizes the proteins or other substances contain comparatively large sized nucleoli. For example, oocytes, neurons, and secretory cells. Oocytes, neurons, and secretory cells. These cells synthesizes actively more compounds. Here, these cells possess large sized nucleoli. So it means the size of the nucleolus is mainly depends on the synthetic activity of the cell. Okay. Coming to the number of nucleolus is depends on the species and number of chromosomes. Usually, one nucleolus will be present per one haploid set of chromosomes. It means the diploid cell contains two nucleoli. Usually one nucleolus is present per one haploid set of chromosomes, so that the diploid cell or diploid nucleus contain two nuclei. The position of the nucleolus is always eccentric, and the nucleoli are formed during mitotic telophase in association with the nuclear organizer, that is NO, which represents the secondary construction of the chromosome. The chromosomes which are associated with nucleolus are called nucleolar chromosomes. Nucleoli are formed during the mitotic telophase in association with the nuclear organizer, that is NO, which represents the secondary construction of the chromosomes. Nuclear organizer consists of genes for 18S, 5.8S, and 28S RNAs. In the region of nuclear organizer, the genes present in this region are 18S, 5.8S, and 28S RNAs. The genes which are present in NO region produce rRNAs, 18S RNA, 5.S rRNA, and 28S rRNA. And the fourth type of RNA, that is 5S rRNA, occurs outside of the nuclear organizer region. The gene which produce 5S rRNA present outside of the nuclear organizer region, and the genes which produce 18S rRNA, 5.8S rRNA, and 28S rRNA are present inside the NO region or nuclear organizer region. And coming to the structure of the nucleolus, nucleolus do not possess a membrane. It means nucleolus do not bounded by a membrane, but calcium ions are supposed to maintain its intact organization. Nucleolus is not surrounded or not bounded by a membrane, but the intact organization is maintained by calcium ions. So that's the reason why the nucleolus appear in spherical shape. Nucleolus exhibits three regions if you observe under electron microscope. They are fibrillar center. Nucleolus exhibits three regions. So one is fibrillar center, second one is granular cortex, and third one is amorphous matrix, When you observe the nucleolus under electron microscope, it exhibits three different regions. They are fibrillar center, granular cortex, second one, and third one is amorphous matrix. Coming to the fibrillar center, it is the innermost region of the nucleolus. Fibrillar center is the innermost region of the nucleolus. It comprises of 5 filaments having 5 to 10 nanometers in diameter and several hundred angstroms long. The fibrillar center comprises 5 filaments. These 5 filaments are having 5 to 10 nanometers in diameter and several hundred angstroms long. These fibrils formed rRNAs and ribonucleoproteins. The fibrils which are present in the fibrillar center formed rRNAs, ribosomal RNAs, and ribonucleoproteins. Coming to the second region, that is granular cortex. This is the outermost region of the nucleolus. Granular cortex is the outermost region of the nucleolus where processing and maturation of pre-ribosomal particles occur. In the granular cortex region, processing and maturation of pre-ribosomal particles are taking place. And third region is that amorphous matrix. This is the proteinaceous ground substance in which granules and fibrils remain suspended. Amorphous matrix is the proteinaceous ground substance in which granules and fibrils remain suspended. If you see the structure of the nucleolus do not possess a membrane, which is not surrounded by a membrane. But the intact organization of the nucleolus is maintained by the calcium ions. If you observe the electron microscope, nucleolus exhibits three distinct regions. They are fibrillar center, granular cortex, and amorphous matrix. Fibrillar center is the innermost region of the nucleolus. It comprises of 5 filaments. Each filament measures 5 to 10 nanometers in diameter and several hundred angstroms long. These fibrils formed ribosomal RNAs, they are rRNAs, and ribonucleoproteins, RNPs. Second one, granular cortex region. This is the outermost region of the nucleolus where processing and maturation of pre-ribosomal particles are taking place. Third region is amorphous matrix. This is the proteinaceous ground substance in which granules and fibrils remain suspended. Coming to the functions of nucleolus. The first function is that the main function of the nucleolus is the biogenesis of ribosomes or synthesis of ribosomes. Ribosomes are produced in nucleolus. Second function is it is one of the main active centers of RNA synthesis. Particularly rRNAs are produced in the nucleolus. And nucleolus plays a significant role in cell division. During prophase, the nucleolus disappears, and at the end of telophase, the nucleolus disappears. Nucleolus play an important role or significant role in the cell division. So these are the three main functions of nucleolus. Nucleolus synthesizes ribosomes and produces or synthesizes RNA, and it plays a significant role in the cell division. And next, coming to the functions of nucleus. Nucleus is one of the important cell organelle, which is the heart of the cell, as I said earlier at the beginning of the topic. Nucleus perform two important and main functions. One is metabolism and the second one is heredity. All the metabolisms of cell are under control of nucleus. Nucleus controls all the metabolisms and functions of different cell organelles. Functions of different cell organelles and metabolisms of different cell organelles are under control of nucleus. For example, if you take photosynthesis, respiration, and protein synthesis, lipid metabolism, and so on. All these metabolisms are under control of nucleus. And coming to the second function, that is heredity. Heredity means the transformation of traits or characters from parents to offspring is known as heredity. The transformation of traits or characters from parents to offsprings is known as heredity. Heredity is under control of nucleus. Actually, the heredity is under control of genes. These genes are present on DNA. DNA present in the form of chromatin in the nucleoplasm. So these heterochromatin which is present inside the nucleus. That's the reason why heredity is under control of nucleus. So these are the two important functions which are under control of nucleus. So that's the reason why nucleus is considered as heart of cell or brain of the cell. Okay, with this I conclude my topic here, and in my next video I am going to explain the ultrastructure of the nuclear pore complexes, which is also very important. And I would like to thank all the viewers who are patiently watching my video and my presentation. Thank you one and all, and we will meet in my next video. Thank you.